The use of animals and other biological resources is critical to many scientific studies. However, researchers, when they try to repeat a colleague's study, often have trouble achieving similar results. One reason is because of how resources are cited in scientific journals. Biological resources come in all varieties, many types of wild-type, mutant, and genetically modified animals, for example, or wide range of biomaterials, like cell lines, oligonucleotides, or antibodies. When researchers write about their work, they commonly refer to such biological resources using nicknames or generic terms rather than specifying the exact animal strains or biomaterials they used and which repositories or sources they got them from. Think of a study's methods section like a recipe. Rather than the recipe calling for yogurt, wouldn't it be more helpful if the recipe specified exactly what type of yogurt and where to buy it? That way, you get the same delicious result each time you make the dish. Not being precise when citing biological resources leads to three main challenges for scientific research and discovery. First, reproducibility, which is the ability of researchers to achieve similar results when they repeat someone else's study. Second, efficiency, which is improved when research resources are visible and accessible to others. And third, connectivity, which is the ability of researchers to identify colleagues with specific knowledge and expertise. The Research Resource Identifiers Project, or RRID initiative, helps solve these challenges. The initiative is based on a concept called an RRID, which is a unique identifier given to a biological resource. An RRID looks like this. It begins with RRID and includes other identifying information like the strain and source of the resource. A resource's RRID will never change. By labeling a research resource with an RRID, researchers can look up information on the resource as well as find all publications that reference that RRID. This increases transparency in the scientific process and makes it easier to find, report, and track information about resources. For example, this paper uses a more generic description and a nickname to describe the mouse resources it used. An issue arises when trying to search for the mouse by the nickname EY217. Two different mouse models are found. On the other hand, this paper in the journal Cell notes the full name, the source, and the specific RRID for the mice used. Antibodies and other resources also are identified using the RRID. Which information would be more useful if you were trying to repeat the study? The RRID initiative also offers a web-based search portal, rrid.site, that can be used either to find a resource of interest or to register a resource for others to find. Let's take a look at an example. At rrid.site, navigate to Find Organisms and type in JAK3. This search will retrieve a list of all available mouse resources that have a JAK3 genetic mutation. The results page can be viewed in Snippet View or in Table View. Searches can be narrowed further. For example, adding MMRRC will retrieve a list of available mouse resources at the Mutant Mouse Resource and Research Center with a JAK3 mutation. To register a resource, scroll down to the bottom of the main page and click on Add a Resource. Registering a resource for others to find will increase the number of resources tagged with an RRID and ultimately help facilitate reproducibility, efficiency, and connectivity of research. Not only have many researchers been adopting RRIDs, but many journals and publishers now require them. The NIH Office of Research Infrastructure Programs, along with several NIH institutes, is supporting the RRID initiative to provide the benefits of this system to the biomedical community. With RRIDs in place, resource providers, funders, institutions, and others can better track resources usage and impact. Here, you can see how the use of RRIDs has grown since their implementation in 2014 in comparison with other resource naming approaches. In navy in this figure is the growing number of papers that cite resources using RRIDs. The RRID initiative is part of a comprehensive solution to the growing demand for transparent and rigorous research. Using RRIDs saves researchers time in the long run, and it is easy to get on board. Visit rrid.site to learn more and start using this important tool today. We welcome any questions and feedback at rii-help@scicrunch.org.